So this, as we're looking at the mitochondria, two things are going to come into the mitochondria. Three things, actually. There are more than three things, but two or three things initially. You're going to have pyruvate, which is coming in from glycolysis, and it's going to go into the mitochondria. You have oxygen gas that either is coming from a chloroplast in a plant or through the stomata in a plant, or it's coming in, if it's an animal, it's coming in through the lungs and diffusing into the blood, then diffusing into the cells, and finally diffusing into the mitochondria. So oxygen comes in as well. So once oxygen is not needed in the first thing that happens to pyruvate, as soon as pyruvate enters, let's take a look at what happens. As soon as it enters the mitochondria, and pyruvate's going to enter the mitochondria here. So this is what's happening in the mitochondria, in the intermembrane space, right? You're going to produce this, pyruvate's going to quickly be turned using CoA. It's going to be turned into acetyl-CoA. So pyruvate is turned into acetyl-CoA. As soon as it does that, as soon as you add CoA to the pyruvate, it becomes acetyl-CoA. What happens is you quickly start to lose CO2. So you start to lose CO2, and carbon dioxide is exhaled. So this is where your CO2, this is where finally the carbon that was part of that sugar in that candy bar you ate or that shake you drank or what have you, this is where that carbon's going. It's going into CO2 and it's leaving your body. So this is leaving. This is going out of the body. So the carbon is destroyed there. One of the carbons is destroyed. Then what happens is it continues the process, the cycle, what's called the citric acid cycle, also known as, probably more people know it as the Krebs cycle. The Krebs or citric acid cycle is going to take, and they call it citric acid cycle because citrate is the first step. Does all these changes, which we don't need to look at all the changes. You don't need to know any of these molecules and how they change. If you were taking a college class, you would want to look at this and see. If it was a biochemistry class or a medical school exam, you might want to know where NADH is made, where QH2 is made. These are regulatory steps, right? GDP being made into GTP. GTP is just another energy molecule like ATP. It's just one of the nucleotides, right? You're going to want to know where ATP is used. You're going to want to know where NADH is made. But in this case, we're not concerned with all these steps. I just want you to understand that what you're responsible for is understanding that in the Krebs cycle, which is where pyruvate goes if oxygen is available into the mitochondria, pyruvate is then broken down into carbon dioxide that leaves the body. So the Krebs cycle, then, let's go ahead and take a tally of it. Let's go ahead and mark where we're producing things. Here we're producing CO2 and NADH plus H+. So those are hydrogen ions. We're going to use those. Here's NADH and H+. Here's NADH and another carbon dioxide. And here's another carbon dioxide. So we lost one carbon dioxide here, another one here, and a third one here. And then here's a GTP. Oops.
You got a ruler?
Here's a oh man, what's this? Sorry about that. Here's a GTP being made. And here's an NADH being made. So those are all where we're gaining something. We're gaining something. Oh, and by the way, we're also gaining it says a Q. We're getting FADH2. That is another electron carrier. It's like NADH. And so when we look at what we're gaining here, those are all the things we're gaining. And the CO2, the carbon dioxide, is going to come out. Carbon dioxide is going to come out and it's going to leave the cell here. It's going to come out and leave the cell here. It's going to come out, obviously leave the mitochondria and then leave the cell. You exhale it. Every time you exhale, you're exhaling the carbons that came from the sugar. Now that's for every pyruvate molecule, and there's 2 pyruvates, right, for every glucose, because remember, they're 3 carbon sugars, so you'll end up making 6 carbon dioxides for every sugar, or 3 carbon dioxides for every pyruvate. Then you end up, now that's all leaving the cell, leaving at least the mitochondria. If it's in a plant cell, the chloroplast will use it. If it's in an animal cell, you'll exhale it. But it's certainly leaving the mitochondria. All right. So then the next thing you have to consider is that you're going to produce, how many NADHs are you making? You're making 1 here, you're making a second one here, you're making a third one over here, you're making a 4th one here. So you're making 4 NADHs and plus 4 hydrogen ions. So that's being made. That's good. Those are all going to, just like the other NADHs, these are all electrons being carried to, where are the electrons going to be carried to? The electron transport chain. And you're also making 1 FADH2. For every pyruvate, and that's going to electron transport. So these 2 are going to electron transport. All 5 of these are going to electron transport chain. Lastly, you're going to make a GTP. The GTP will be used in the process, but not in the electron transport chain. But you ended up losing, you made 1 GTP, but then you lost 1 ATP. So it kind of evens out. GTP is very similar to ATP, except that instead of adenosine triphosphate, you have guanosine triphosphate. So they're just different nucleotides, but they pretty much even out, and you have a net 0 gain there. So in the ETC, you're getting the NADH from the Krebs cycle. That's the last of the pyruvate. You're getting the NADH from glycolysis in the cytoplasm that's coming into the mitochondria, and you're getting the FADH2. All those are bringing electrons to be used by the electron transport chain. All right. So here you have the electron transport chain. I think you'll see that it's very similar to what we saw in photosynthesis, except obviously we don't have a photon nor a photosystem to deal with. Here, we're just taking the electrons that we got from the citric acid cycle here in the matrix, right? The citric acid or Krebs cycle. I'm going to go ahead and write Krebs again so everybody remembers that they're the same thing. We just looked at the Krebs cycle. You don't need to know the details. Just know that the NADH and FADH2 came from the Krebs cycle. And what happens is that NADH then drops the electron off here at the first step. And every time the electron gets transferred, and by the way, this Q and this CYTC, just like in our activity that we did a week ago, the Q is quinone, and this is cytochrome C. These are the molecules that take them and move them from these hydrogen transporters over to the next electron, right? So these guys, that's quinone and cytochrome C, are carrying the electrons from one protein to the next protein to the next. That electron's energy is used to pull a hydrogen from the low to high. So remember, hydrogen ions are low here, and their hydrogen ions are high out here. So high concentration of hydrogen ions means that you have a low pH, because remember, the relationship between pH and hydrogen ion concentration is opposite, but that also means it's acidic. So relatively speaking, you're more acidic in this space, in this intermembrane space out here. This is more acidic, and the space in the matrix is more basic. All right. All right. So now we have all these hydrogen ions in the intermembrane space. How do we get them? How do we use that? So we're going to look at this as if we had dammed up a river, right? So if we dam up a river, what we find is that on one side, we have a lot of water, obviously, and it's high up. It's a cross-section of a dam, like at Niagara Falls. And then if we open up a hole in the dam, then what happens is water flows out. Well, heck, if we attach a wheel to that, some kind of turbine, we call these turbines, that water will turn the wheel. And the turning of the wheel, this mechanical energy, I can use it to run electricity. It can make electricity, right? So we can make electricity. That we can run to your house. So Niagara Falls actually uses this technology to make electricity. Well, we can use this technology in our cells. In fact, we do. We have a high hydrogen ion concentration, like having a lot of water on one side of a dam. Then we create a hole that allows the water to flow, which allows us to turn. Well, this high hydrogen ion concentration, we have a hole in this thing called ATP synthetase, which we've seen before again. Which turns round and round as the hydrogen ions flow through, and every time it turns, it makes ATP. It makes a lot of ATP. So we make 36 ATPs here. So that's a lot of ATP for every glucose molecule. So you can see that we get a lot out of this electron transport chain. We use the chemiosmotic gradient of this hydrogen ion concentration to drive to give the ATP synthetase the energy it needs to be able to allow it to turn, to have hydrogen flow through, and that allows ADP to be combined with ATP, allows us to get the energy to bond a phosphate onto ADP, making this high energy molecule that we can use all over the cell. So that's important. This is in the mitochondria. This is the matrix, the space inside the inner membrane. This is the inner membrane space that's just outside it. And these are the carriers. Do you need to do let's see, do you need any other details? Oh, well, one more thing I should mention. Oxygen, when the electron finally gets to the last the last protein in the chain, the electron transport chain. We need to do something with that electron. Otherwise, it'll be like a car stopping on the highway. You're going to have the highway backed up. So we got to get rid of this electron. Got to keep this electron moving. And the way we do it is we take it and we recombine it with oxygen. Remember, we got the electron originally in photosynthesis. Water was broken. We got our electrons from water in photosynthesis. But now we're using oxygen to make water. So this is why it's such a beautiful cycle. The cellular respiration makes water, where photosynthesis breaks water apart. And so oxygen is the final electron acceptor in this chain, and this water then gets urinated out, or if it's in a plant, it's used in the chloroplast to break apart so the electrons can be moved from one place to the other. Water is actually the facilitator of that. So oxygen is the final electron acceptor. That's the only reason you breathe, is so that you can take up those electrons. Breathe oxygen is so you can pick up these electrons and keep this ATP flowing, and also to get rid of carbon dioxide that you made in the Krebs cycle, or right before the Krebs cycle, and then in the Krebs cycle. So I hope that helped you understand the 3 processes you need to know. You don't need to know any of the enzymes. You should know what quinone and cytochrome C are. You should know what ATP is, obviously. You should know the general outline of the mitochondria, and you should know ATP synthase. But all those other enzymes, all those other steps are not what's key. What's key is that you understand the process, you understand the flow of the molecules, the decision point when you make the pyruvate. Are you going to do oxidative phosphorylation? This is called oxidative phosphorylation. All right, so I'm going to go ahead and write that word down because it's big, but it's oxidative phosphorylation.
This one right like that one too. This one right like that. This one right like that. Do the red. This one, no, the red baby.
So oxidative phosphorylation is just a fancy word for saying electron transport chain. Why do they call it that? Because we're adding phosphate at the ATP, right? Look at this. There's phosphate there. Adding it to ADP, so that's phosphorylation, adding phosphate, phospho. And we're transporting electrons from one carrier to the next, driving the creation of the hydrogen ion concentration high and low, concentration differences, the hydrogen ion concentration gradient. And that allows us then to make this whole thing work. The reason this is important, because it makes us a lot of ATP really quickly. We're doing it at substrate level phosphorylation. That's the alternative. Substrate level phosphorylation is done one enzyme, one substrate, putting one phosphate on one enzyme putting a phosphate on one substrate at a time. So this is actually much slower. It's slower, but it's also more targeted. I'll tell you what, I'll let you guys present it on Monday. And along with your quiz questions. Get your work done. I need you all, the reason I want you to present is because you all need to know all of these signaling mechanisms. Are you clear? I don't know what they're going to give you on the AP test. I don't know if they're going to give you tyrosine kinase or they're going to give you G. Your test is on Tuesday. Phosphorylation is slower, more targeted. It's slower but more targeted. Where electron transport chain is like a factory, the substrate level phosphorylation is like an artisan or someone making a chair at a time, where oxidative phosphorylation is like a factory making hundreds of chairs in the same amount of time. So the relationship is there. They're both phosphorylating. They're both making ATP. Not all phosphorylation is making ATP because kinases can phosphorylate other molecules. But when ATP is phosphorylated, you're making energy. One makes energy a lot, really quickly, oxidative phosphorylation, the electron transport chain, or the substrate level phosphorylation, slower and more targeted. All right, I hope you understand that. I hope you get the key points. Also do the chapter reading and the activities, and I think you'll be fine.